When things need to move in and out of a cell, they tend to have to use channels, except for the small nonpolar lipid soluble things. Um, so, with that, the channels have various properties that go along with them to allow them to move things in a certain fashion. So the first thing that we talk about when we talk about channel dynamics is their specificity. Okay? So specificity means that they are specific to one thing or another. So if I have a membrane here and I am going to put a membrane channel on it, so we'll just sort of make this one like a tube looking channel. And this channel is responsible for moving, let's call it sodium. So sodium can move through this channel. Okay? But let's say chloride tries to come in and tries to move through this channel. It's blocked. Okay? So how is it possible that we can have this level of specificity with these different channels? If we sort of blow the channel up a little bit and we take a look at the inside, like so, what would it be like, right, if we took inside of this channel and we put a whole bunch of negative charge here on this inside of this channel, right? And we tried to move a thing like chloride that has a negative charge through this channel. These negative charges are going to repel that chloride and block its movement, right? So the composition of the protein, the amino acids that make up the protein, can dictate its specificity and help dictate its specificity. Aside from that, other things like size, shape, can also dictate whether or not a channel can move one thing or another. So channels have a high level of specificity. Okay? On top of that, these protein channels, let's say we are looking at a channel and that channel is responsible for moving sodium. So we'll make our blue sodium channel again, like so. Okay, and it moves some sodium through the channel. Okay, well, what happens if I put another sodium out here? What sodium can move through the channel? Well, what if I put another one, and another one, and another one, and another one, and another one, right? How many can move through that channel, right? So we tend to talk about channels and their transport rate. So let's look at this a little bit differently. See if we can't use some analogous examples. Okay, so I live in Ohio, and one of the major highways that runs through Ohio is I-75. I'm going to take I-75, and I tend to drive along this highway quite frequently, and this highway runs north and south throughout the state, and it's a multi-lane highway. Okay, now because I am in Ohio, one of the things you might know about Ohio is that all throughout the state, especially on the highways, are the red and blues, policemen, the cops. Okay, and they sit along this highway. Okay, and they sit along this highway because they know that people speed on this highway. Okay, and so they try to catch the speeders on this highway. Now, if these cops, let's say that the cops are able to pull over 10 cars per hour. That's what they can pull over. 10 cars per hour. Okay? And let's, for the sake of my argument, assume that all the cars that come down this road speed. Okay? And so what happens if I spin and send one car down this road? Cops are going to pull over one car. What if I send two? Two cars get pulled over. What if I send 3 cars per hour? 3 cars get pulled over. What about 4? And you can see where this is going. But what happens when I get to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 cars? Well, 10 of them, 5, 6, 7, 8, 9, 10 of them get pulled over. What happens to the other 3? They sneak by, right? Because the cops get saturated with cars that they can pull over. That is more cars passing per hour than they can pull over, right? So the same thing sort of happens when we look at our membrane channels, right? And so if we look at a graph here and we put the concentration on our x-axis of the solute, and over here on the y we put the rate of transport, and the graph doesn't really matter in terms of the scale, right? But if we look at our concentration or rate of transport, as we increase the concentration, the rate of transport goes up in a pretty linear fashion, right? So as we increase one more molecule, we can transport one more molecule. But there's a point up here where those transporters become saturated. There's more molecules than what they can move, right? And so as we approach that sort of line there, we sort of top off, right? And we hit what is called the transport maximum, often abbreviated TM, or some people will call this Tmax, okay? And it's at this point that those channels are said to be saturated, okay? So with that, right, we can only transport as many things as possible. Now, if we look at this, right, if we were to continue at a true 1-to-1 ratio, we would come up and then come up here and then eventually level off and come straight across. Okay, so this here, right, is sort of like ideal function, right? Whereas in white, we have what is reality. And so because of that, you get this like curve here. See that curve? That curve is known as the splay. Okay, so you'll see that when looking at transport rates is what that splay is. Okay, and that's sort of dictated by the free energy that is around with those transporters. Okay, so if this is the case, right, and I want to transport more things, how do I go about doing this? Right? What if I wanted to pull over more cars? Because we found, like, well, our cops are really busy. We can't pull over any more cars than what we're already pulling over. Well, what we do, right? Ohio's pretty good at this. If you've ever driven through Ohio, is that they put more cops, right? So if I want to move more things, and I want to increase my transport maximum up here, right, I have to add more channels, right? Or add more open channels. So what happens is we said we increase our permeability to whatever it is that we're trying to transport, right? So if we add more channels or open more channels, we are increasing our permeability, which increases our transport maximum, or the rate at which we can transport things. Okay? And so that will become important later on as we talk about the results or the product of moving things in and out of a cell, causing these cells to do a variety of different things. You'll see this come up again when we talk about glucose transport in the kidney. You'll see this come up when we talk about action potentials in neurons, right? So this is an important sort of concept to understand. So with that, we will see you next time.